Okay, on to something that is really, really, really important because we're using this more and more in the crime scene investigations. It is DNA. So, DNA is obviously one of the best ways to identify who did what, when, where, all that good stuff, because DNA can be found in many different things. So before we go into that, let's talk a little bit about DNA and its kind of its structure. So basically, you guys should have learned this in biology, the double helix, okay, and they're made up of 4 nitrogen bases. So you have adenine, thymine, guanine, and cytosine, always pairing with the same 2. So you see that there's G and C, so guanine and cytosine are always going to pair together, and adenine and thymine are always going to pair together. So these are called base pairs. A gene is a unit of heredity. Basically, it's just a segment of DNA. Okay, so you can't. There's so many genes found in this one, structure of DNA. So, anyways, that's a little bit about the DNA just in general, which you should already have a good background on it, as well as this stuff. This is about the replication. Okay, so remember, if you remember, basically, in interphase of mitosis, you have the DNA that replicates. Okay, so what's happening is you have it kind of unzip, and then you have these different things that come in here, DNA polymerase, things that basically bind to that new strand and make kind of an exact replica. Okay, so there you can read through this information here, some more details, but understand a little bit about how DNA reproduces itself. All right, so here's a little bit about chromosomes. Humans always have 23 chromosomes, okay, 23 pairs, sorry. So we actually have 46 chromosomes, but 23 pairs. And they're numbered from 1 to basically 23, 23 being the sex chromosome. So in a male would always have X and Y, and a female would always have XX, so 2 same of the X chromosomes. But basically, starting with chromosome number 1, you have the largest chromosome that so many genes are found on, okay? I think down here, yeah, it says 2,800 genes on basically this first chromosome. And then obviously, as you get to these smaller chromosomes, there are going to be less genes on there. But these are kind of what our chromosomes look like that just house all of that DNA and, again, those genes as well. So DNA profiling, this was a big thing, and I believe it was like 1980s that this really came about. That basically you can collect DNA, and you can collect it from different places, the semen, the saliva, hair follicle, body cells, skin, and blood. Those are the different places that you can actually gather evidence, okay, from maybe a suspect or victim and so forth, okay? And I'll read through these bullet points because these are pretty important. It says forensic scientists usually collect DNA with a cotton swab or by cutting off any stains from clothing or fabric. While collecting evidence, forensic scientists wear gloves, a mask, use disposable instruments, and avoid talking or touching parts of the body. You don't want to contaminate that. The sample should be air dried, okay? That's a big thing because basically if it's put in something like plastic, it could being that it's a little moist, it could form mold, and that would contaminate that DNA as well. And then if the samples can't be air dried, they should be frozen. So that's about collecting it, okay? Now a big portion of this little unit is on what they do with it. I'm not going to go over all of those details specifically. I will probably lose you. I want you guys to know that this particular unit, this little, the DNA portion, is going to have to sink in over time because there's a lot of different, first of all, a lot of different names to what they do with all the DNA, but also, the numbers and what everything means. Let it kind of sink in. The lab that you guys, if you choose to do the DNA lab, you'll be amazed as it will be a little more intuitive than you would think. But don't let this, if this doesn't totally sink in and you don't really understand everything about it, that's okay. It'll start to make sense, especially if you really enter in this field. You'll learn a lot more about this, okay? So anyways, DNA profiling. Again, profiling, taking DNA, figuring out who it belongs to. But basically, there's one particular type called the restriction fragment length polymorphism, okay, the RFLP. And basically what it does is it takes this DNA from, say, a blood stain, and you add something to it, this restrictive enzyme that basically cleaves the DNA into different segments that can be looked at in a little bit more closer detail. So that's what that does, the big long word there. Gel electrophoresis. Okay, I'm going to read this part and go over this with you and show you these pictures, and you can obviously do this yourself as well. But gel electrophoresis is definitely an important technique in separating the DNA fragments. Okay, this photograph, a technician is going to add samples to these wells. You can't really see them that well, but and it has this gel matrix. Okay, so that's the first thing. Samples containing fragments of DNA are loaded into the wells at one end of the gel plate. So right here are your samples. They're going to be added into these, basically where there are gel underneath there. And what's going to happen is an electric current is going to be applied to it, and it's going to basically move that DNA towards the positive electrode. So it starts at the negative. These are where the wells that you added the sample to, but the positive electrode, basically, because of that electric current, it's going to bring that DNA close. But it's going to have a specific pattern based on that person's DNA. So the DNA fragments will separate because of lower mass fragments, and they move more quickly through the gel. So they will separate, okay? And then ultimately you get a pattern. So this pattern is produced by the electrophoresis. It can be matched to DNA from a crime scene. So here's the crime scene DNA, and here are the 3 suspects. So you can see that obviously suspect number 2 is matching up with that. So DNA electrophoresis is a big, big deal. It helps a lot in figuring out, who these DNA samples belong to. All right, so again, here's more ways to do this. Okay, talking about the RFLP that we discussed a little bit, 2 slides ago or whatever, basically required large amounts of DNA. That was a really tough thing because sometimes you didn't have large amounts. So one great thing is basically this PCR, okay, polymerase chain reaction, uses DNA polymerase to basically copy that DNA and make more of it, which is always a good thing. If you have more DNA, you can do more testing. So the PCR, which again is the polymerase chain reaction, basically aids in creating more DNA from what is in existence. Okay, so that's a pretty good thing, right? All right, and this is just showing kind of a picture of, here's kind of what you have, and you can amplify it to this many copies. So, yay! All right, now, modern DNA profiling depends on variations in the non-coding sequence. That's a big deal because the way that DNA profiling happens today actually goes by the non-coding sequence. And what that means, and I'm going to read this to you guys, and again, let this stuff sink in. The non-coding portion of DNA contains variations between individuals. This fact is the basis for modern DNA profiling. Specifically, molecular biologists found short repeating sequences of 2 to 5 base pairs, or sometimes even more of those repeating, in the non-coding sections of DNA. Such a sequence is called a short tandem repeat. So you'll see this STR in many different places. So just know what STR is. It's called a short tandem repeat, meaning we're talking about those non-coding places where you basically have these repeating sequences of the non-coding DNA, okay? So molecular biologists found the following. Hundreds of STRs exist throughout the entire DNA. The on-screen examples show one, ATG. You can see these are non-coding, but they're repeating, okay? This one individual has 7 repeating units. This individual too has 8, okay? And then the last little thing is the number of STRs varies among individuals, okay? So you can see obviously the difference in that. Now typically when there is some DNA profiling going on, they, and in the United States, will take 13 of these STRs to look at. And then it actually, when you think about it, you're not looking at the entire DNA strand, you're taking just little snippets of it, but these are very specific to individuals, so it's a quick, easy way to basically profile that DNA. Okay, so hopefully that makes a little bit of sense, because that's kind of what you're going to be doing in the lab. So again, like I said, DNA profiling in the United States compares 13 of those standard STRs to form a profile, okay? Alrighty, now STRs have alleles. Here's another portion that you need to understand for your lab. That basically, if you guys remember from biology, you have genes that contain alleles. So like say for instance you have the gene for brown hair, could be big B, or big B, little b. Those are the 2 alleles. Well, same with these STRs, okay? So basically when we talk about STRs, it can still be homozygous, okay? Which is if the number of the repeat units at the STR locus is the same, for both alleles, it's homozygous. Remember, homo same, hetero different. So if it's heterozygous, the number of repeat units are going to be different at each allele. Okay, so hopefully that kind of brings back some memories from biology, and you guys can kind of remember that when you go to do your lab, because it'll talk about the homozygous versus the heterozygous. Okay, so here's just an example of using the fluorescent light to kind of view this DNA in a gel electrophoresis. It's kind of a cool looking thing. But if you guys consider taking more classes in college, you will actually be doing this, so it's kind of cool. But you can really kind of tell what's going on in this gel phoresis with that, the UV light. Okay, and then I'm not going to go over too much more. I want you guys to be able to read through some of this stuff. But again, there's some different things. It says the modern DNA profiling can use capillary electrophoresis. It's kind of the same principle as the gel electrophoresis, but basically, right as it says here, the fragments are going to move along thin capillary tubes filled with a polymer. Okay, a window in the tube allows the detector to detect fluorescent STR fragments as they pass. So kind of similar to the gel, but a different way to kind of capture that information. All right, and then the last thing that you guys are going to be really looking at for the lab is the electropherograms. Okay, electropherograms, and I'll show you guys. I believe there's an image next. But basically, it's showing these STRs, okay? It's kind of a record of them. So basically, these forensic scientists can look at these electropherograms and really start to pull out some information and see what is matching up with maybe the actual crime scene evidence, okay, for the DNA that's extracted. So here's an electropherogram. I want you guys to actually click on this. If you click listen, you will be able to just kind of listen and watch as it takes you through each of these components here. Okay, so basically 1 through 9 will give you a good background on what to do and how to use this when you go to do your lab. Alrighty, that's pretty much it that I'm going to go over. You'll see something like this in your lab as well. So basically you can look at some of the different locus, the STR locus, and kind of the evidence sample, and you can compare them again with the suspects. So kind of important because obviously this is what forensic scientists are looking at when they are really analyzing the DNA to figure out who that sample belongs to. So I hope I didn't overwhelm you guys with this. This is going to be one of those lessons that is going to be kind of like, whoa, I'm not sure if I'm understanding anything that she's saying, but I promise this is probably one of the more harder concepts. Just because you're dealing with, think about it, DNA has so much information. I mean, it can tell so much about a person, but therefore we're actually using such a tiny sample of it to try and identify who it is. So you can imagine that that's going to be a little bit more complicated than, say, finding something, a fingerprint, so to speak. Okay? So anyways, hopefully that gives you just a start. Keep reading through this, and it'll all start to come together, I promise. All right, bye-bye.